Now we'll focus on the amino acids. And we're going to specifically focus on the polarity of the R groups, that attached group on the amino acid. As mentioned previously, there are 4 parts to the amino acid. There's the amino group, the carboxylic acid group, the hydrogen, and the R. Now the R group is something that changes. Each amino acid has a different R group, and the R group determines how it behaves in the body. So we're going to focus on each of the different R groups and talk about how they interact with each other, dependent upon their structure. The first set we'll look at are the nonpolar ones. Nonpolar R groups are ones that are composed almost essentially of carbon and hydrogen. There are 2 exceptions. There's here one with a nitrogen in it and one with a sulfur. The one with the nitrogen still has a very large carbon content. The one with the sulfur does not have a sulfur with a hydrogen, and that helps to keep these hydrophobic. When we talk about something being hydrophobic, that means that it hates water. And since it hates water, it tends to hide and be on the inside or interior of the protein, where it can avoid water, rather than the outside or exterior of the protein, where it would have to interact with water. Our second classification of R groups are the ones that are polar. Now, polar ones generally have an OH, an NH, or an SH easily found in the majority of the structure. There is one major exception, and that is this guy here with the hydrogen only. The reason this one is considered hydrophilic is because of the C double bond O and the nitrogen that are found in the main chain of the amino acid. So those allow it to become polar, that the overall molecule is polar. There's not a lot of other hydrocarbon to cancel out that highly polar section. At any rate, we'll notice that they all have OHs and NHs, and those make it polar. Now, if it's polar, that means it likes water, and so we expect to find it on the outside or exterior of the protein, where it can interact with the water. And our last grouping, these are hydrophilic also, are the ones that are ionic. And so you'll see that they all have a charge. So we have some negative charges or positive charges, depending on the amino acid you're talking about. But because it is charged, these groups like to be in water. They're hydrophilic. And since they like water, we expect to find them on the outside surface or the exterior of the protein, where they can be in contact with water. When we look at polarity of amino acids, we can see they basically range from ionic, which is very, 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 very polar, all the way down to nonpolar. And so this ranking we can see on this chart, we're going to go from 60 down to 0. And looking at this chart, we can see that something above 50 we would consider ionic or very polar. If we see something yellowish orange, we're going to see that those are what we consider very polar, and those are around 40. If we go down to 30, those are sort of getting close to being nonpolar. They're not quite nonpolar, but they're close. That's around 30. If you go all the way down to 20 and below, you're going to consider those to be nonpolar, and those are in our blue range. So looking at this image, you can see that the red ones, something around 50-ish, 60-ish, are on the very outside parts of the protein. As you follow through, you see some yellows, kind of lower polarity, but still considered very polar. Those are on the outside also. And then we move into the ones that are green, and you'll see that they're kind of borderline. Some of them are on the outside, some are on the inside. They're sort of a little bit polar, but not very, very polar. So we see them mixed in the middle here, where they're kind of on the surface, but also sort of on the inside. Kind of borderline on there. But you'll notice that on these coils, the yellower parts, the ones that are more polar, are towards the outside, even on the coil, and that the ones that are more blue are towards the inside. It gives us a kind of a buffer zone where the polar stuff and the nonpolar stuff can be close to each other, because we have this sort of semi-polar stuff in the middle, the green areas. Then again, you'll see in the very center, we'll start to see the darker blues. As you move into the blues, you see they're much more in the center interior of the protein.